8 experiments that changed the world. Our modern world, with all its incredible tech and deep understanding of the universe, wasn't a given. It was built piece by piece on a handful of crucial discoveries, and these weren't just eureka moments. They were the result of strange, difficult, and often baffling experiments. But what if they never happened? What if a few key experiments failed or their results were ignored? We're going to take you inside 8 history-making experiments that unlock the secrets of our universe and literally built the reality we live in today. From the very nature of matter to the code of life itself, these are the stories of how we figured out what and who we really are. 1. Rutherford's gold foil experiment, unveiling the atomic nucleus. At the dawn of the 20th century, our picture of the atom was fuzzy, to put it mildly. The best idea anyone had was the plum pudding model. It imagined the atom as a soft blob of positive charge with tiny negative electrons dotted throughout like plums in a pudding. It was a nice, neat idea, and it was about to get completely demolished. Physicist Ernest Rutherford, along with his team, devised an experiment that seemed pretty straightforward. They would fire tiny, positively charged alpha particles at a sheet of gold foil, so thin it was almost see-through. They figured the particles would zip right through like bullets through a fog. And most of them did. But what they saw next was, in Rutherford's own words, as incredible as if you had fired a 15-inch shell at a piece of tissue paper, and it came back and hit you. A tiny fraction of the particles were deflected at huge angles, and some even bounced straight back. This could only mean one thing. The atom's positive charge and all its mass weren't spread out at all. They were crammed into an impossibly small, dense center. Rutherford had discovered the atomic nucleus. That single experiment threw out the plum pudding model and gave us the picture of the atom we know today. A dense nucleus with electrons orbiting it. This was the launch pad for nuclear physics, quantum mechanics, and the atomic age itself. Every nuclear power plant, every medical scan, and our entire understanding of matter started right here, with a few unexpected flickers on a screen. 2. The Michelson-Morley experiment, the failure that broke physics. Imagine the entire universe is filled with an invisible, undetectable substance. For 19th century physicists, this wasn't science fiction. They called it the luminiferous ether, and they believed it was the medium that light waves had to travel through, just like sound travels through air. Finding this ether became a scientific obsession. In 1887, Albert Michelson and Edward Morley built a brilliant device to do just that. Their interferometer would split a beam of light, send the two halves on a race down perpendicular paths, and then bring them back together. The idea was that if Earth was moving through this ether, one beam would be fighting an ether wind while the other would be going across it. This should create a tiny delay, a telltale shimmer when the beams recombined. They set up their experiment on a pool of mercury so they could rotate it, hunting for the ether from any direction. They ran it at different times of day, different times of the year, and they found absolutely nothing. No shift, no delay. The speed of light was stubbornly, maddeningly the same in every single direction. The Michelson-Morley experiment is often called the most famous failed experiment in history. But its failure was its genius. The null result triggered a crisis in physics. It meant there was no absolute frame of reference, an idea that had been gospel for centuries. This shocking result was the clue a young Albert Einstein needed. Instead of trying to save the ether, he took the result at face value and proposed that the speed of light is a universal constant. It was a radical idea that became the bedrock of his theory of special relativity, forever changing how we see space, time, and gravity. 3. Millikan's oil drop experiment, weighing the electron. By the early 1900s, scientists knew the electron existed and that it had a negative charge. But they had a huge question. Is electricity a continuous fluid, or does it come in tiny individual packets? In other words, is there a single, smallest unit of electric charge? American physicist Robert Millikan decided to find out. His setup was beautifully simple. 2 metal plates with an electric field between them. He'd spray a fine mist of oil into the chamber, and the tiny droplets would pick up static electricity as they fell. Using a microscope, Millikan could watch a single oil drop fall under gravity. Then he'd flip on the electric field, creating an upward force. By tweaking the voltage, he could get a droplet to just hang there in midair, perfectly balancing gravity's pull with the electric push. By measuring the field and knowing the droplet's mass, he could calculate the exact charge on that one drop. He did this over and over for hundreds of droplets, and what he found was a revelation. The charge on the droplets wasn't random at all. It was always a multiple of one single tiny value, 1.602 times 10 to the negative 19 coulombs. This was it. The fundamental unit of charge, the charge of a single electron. Millikan had proved that charge is quantized, meaning it comes in distinct packets. Although his methods were later criticized for cherry-picking data, his result was incredibly accurate and won him a Nobel Prize. That number became a crucial constant in physics, essential for everything from quantum mechanics to the design of every electronic device you own. 4. The Miller-Urey experiment, creating life's building blocks. Where did life come from? It's pretty much the biggest question there is. In the 1950s, a few scientists had a hypothesis. If the early Earth's atmosphere was a primordial soup of simple chemicals, maybe the energy from lightning could have spontaneously cooked up the building blocks of life. It was a cool idea, but just an idea. Then, in 1952, a grad student named Stanley Miller, working with Nobel laureate Harold Urey, decided to test it. They built a contraption of flasks and tubes to mimic the early Earth. They filled it with water for the oceans and a mix of gases they thought made up the atmosphere back then: methane, ammonia, and hydrogen. For lightning, they zapped the gas with electric sparks. They just let the experiment run for a week. By the end, the water had turned a murky brownish red. When Miller analysed what was in that murky water, he was stunned. It was full of amino acids, the fundamental building blocks of proteins which are essential to every living thing we know of. The Miller-Urey experiment was a landmark. It was the first time anyone had shown that the complex molecules of life could form from simple, non-living chemicals, all on their own. Now, our understanding of the early Earth's atmosphere has changed a bit since then, but the core finding is still incredibly powerful. It launched the fields of prebiotic chemistry and astrobiology, and it showed that the stuff of life might not be a one-off miracle, but a natural consequence of chemistry, here on Earth and maybe, just maybe, elsewhere in the universe. 5. The Avery McLeod-McCarty experiment, identifying the stuff of genes. By the 1940s, scientists knew something was being passed from parent to child, a substance that carried the blueprint for life. They called them genes, but they had no clue what they were made of. The main suspect was protein. Proteins are complex and do all sorts of jobs in the cell. DNA, on the other hand, was thought to be a simple, repetitive molecule, way too boring to hold the secrets of life. That idea was shattered by a quiet, careful experiment from Oswald Avery, Colin MacLeod, and Macklin McCarty. They were building on an earlier discovery where a harmless strain of bacteria could be transformed into a killer strain just by mixing it with heat-killed killer bacteria. Something from the dead bacteria was passing on the deadly trait. That something had to be the genetic material. So the team took the dead, deadly bacteria and painstakingly separated all its parts: its fats, its sugars, its proteins, its RNA, and its DNA. Then they systematically tested each one. They took 5 batches of harmless bacteria. To the first, they added just the protein. To the second, just the RNA, and so on. The result was crystal clear. The bacteria that got the protein stayed harmless. The bacteria that got the RNA stayed harmless. But the bacteria that got the purified DNA were transformed. They became deadly, and they passed that trait to their offspring. To be absolutely sure, they added an enzyme that destroys DNA, and the transformation stopped cold. It was conclusive proof. DNA, not protein, carries our genes. The discovery was met with a lot of skepticism at first, but it completely redirected biology. setting the stage for the discovery of the double helix and kicking off the age of molecular genetics. 6. Franklin, Watson, and Crick, revealing the double helix. Once DNA was crowned the genetic material, the race was on to figure out its structure. Scientists knew that if they could see the structure, they could understand how it worked, how it stored information and copied itself. The answer came not from one experiment, but from a collision of brilliant work, shared data, and a bit of controversy. At King's College London, a chemist and X-ray crystallographer named Rosalind Franklin was using her expertise to take pictures of the DNA molecule. By shooting X-rays at tiny DNA fibres, she could capture the diffraction pattern, which held clues to its shape. Franklin was a meticulous scientist, and she produced the clearest images of DNA ever seen. One in particular, Photo 51, showed a stunningly clear X-shaped pattern. To a trained eye, that X screamed helix, a spiral structure. Meanwhile, at Cambridge University, a biologist, James Watson, and a physicist, Francis Crick, were also on the hunt, trying to solve the puzzle by building physical models. Their first few attempts were total failures, but then they were shown Franklin's Photo 51, reportedly without her full permission. That picture, combined with Franklin's own precise measurements, was the final key. Watson and Crick realized DNA wasn't just one helix, but a double helix, like a twisted ladder. This structure immediately explained it all, how DNA could be unzipped down the middle and copied perfectly. Their 1953 paper announcing the discovery changed biology forever. While Franklin's critical role wasn't fully celebrated until much later, her work was absolutely essential to one of the greatest breakthroughs in history: cracking the code of life itself. 7. Pavlov's dogs, conditioning the mind. Not all world-changing experiments involve chemistry labs. Some just need dogs, bells, and a bit of drool. In the 1890s, the Russian physiologist Ivan Pavlov wasn't even studying psychology. He was studying digestion, measuring how much saliva dogs produced when they got their food. But he quickly noticed something odd. The dogs would start salivating before they even saw the food. They'd drool at the sound of the lab assistant's footsteps coming down the hall. Pavlov realized he'd stumbled onto a fundamental type of learning. The dogs were associating a neutral signal, the footsteps, with the food that was about to arrive. Intrigued, Pavlov designed a new experiment. He started ringing a bell right before feeding the dogs. At first, the bell meant nothing, but after doing it over and over, bell then food, the dogs started to connect the two. Soon Pavlov could ring the bell and bring no food at all, and the dogs would still drool. He had discovered what we now call classical conditioning, how a neutral signal can trigger a response after being paired with something that triggers it naturally. It showed that learning can be an automatic, unconscious process. Pavlov's work became the foundation for behaviorism, a major school of thought in psychology. Its principles have had a huge impact, influencing everything from therapy for anxiety, to how we train our pets, to the way advertisers try to sell us products. And it all started by just paying attention to a dog's dinner time. To subscribe to our channel and hit that notification bell for more stories about the science that shaped our world. And let us know in the comments which experiment you think is the most mind-blowing. 8. The Luria-Delbruck experiment. The randomness of evolution. How does evolution actually happen on a microscopic level? Let's say bacteria become resistant to a virus. Does the virus cause the lucky bacteria to mutate and survive? Or do random mutations just happen all the time and the virus simply kills off the unlucky ones? In 1943, biologist Salvador Luria and physicist-turned-biologist Max Delbruck designed a beautiful experiment to solve this puzzle. They started with one big flask of E. coli bacteria that were all vulnerable to a virus. They let the bacteria grow and then spread many samples from this single flask onto petri dishes coated with the virus. As you'd expect, they found roughly the same small number of resistant bacteria on each plate. But then they did something different. They took a few bacteria and used them to start lots of separate, small cultures in different test tubes. They let them all grow to the same size. Then they plated the entire contents of each individual tube onto its own virus-coated dish. If the virus was causing the resistance, then every bacterium would have the same small chance of changing when it met the virus. And so every plate should have about the same small number of survivors. But that's not what they saw. What they saw was wild fluctuation. Some plates had 0 survivors, a few had 1 or 2, and every once in a while a plate would be a jackpot covered in hundreds of resistant colonies. Luria had a flash of insight, supposedly while watching a slot machine. The only way to get a jackpot was if a random mutation for resistance happened by pure chance very early in the growth of that one specific tube, creating millions of resistant descendants before they ever saw a virus. The plates with 0 colonies came from tubes where, by chance, a mutation just never happened. This proved it. Mutations are random and spontaneous, not directed by the environment. This experiment confirmed that Darwin's ideas of random variation and natural selection apply even to microbes, a discovery that is fundamental to how we understand antibiotic resistance today. Conclusion. From the heart of the atom to the blueprint of life, from the fabric of the cosmos to the wiring of our own minds, these 8 experiments are more than just data points. They're monuments to human curiosity. They're stories of people who dared to question the truth, to build bizarre contraptions, and to look at the world in a way no one had before. Each discovery built on the last, weaving together the understanding that makes up our modern reality. They remind us that the world is full of incredible secrets waiting to be unlocked not by a single flash of genius, but by the patient, persistent, and often surprising process of just trying things out. What will we discover next?